Hi, my name is Nate Looker. I'm a soil scientist with the Soil Health Institute. Today I'll be telling you about our work setting soil health targets for 3 cotton-producing soilscapes. The Soil Health Institute is a nonprofit based in North Carolina with a mission of safeguarding and enhancing the vitality and productivity of soil through scientific research and advancement. Much of our work focuses on promoting soil health management systems, which integrate practices such as reduced tillage and no-till, cover crops, and integration of crops in livestock production. We take an interdisciplinary approach to promoting soil health management systems, considering not only the biophysical aspects of quantifying soil health, but also the broader social and economic context in which producers are making decisions. The work that I'm talking about today, about soil health targets, is most directly related to measurement, but also cuts across other aspects of producers' decision making. Soil health targets are an effort to answer the question, how much healthier can my soil get? Soil health targets are conceived as interpretable goals for soil health improvements. These targets are expressed relative to soil health status of soils with similar inherent properties in reference states. They're intended to be a scalable and intuitive complement to existing approaches, such as dynamic soil properties, soil health scoring functions, and the concept of the soil health gap. Let's take a second to clarify what we mean by interpretable goals, soils with similar inherent properties, and reference states. And then we will apply these concepts to cotton-producing soilscapes in Texas and Arkansas. Soil health targets are considered to be interpretable goals because we take diverse soil health metrics, some of which have units that don't really have an intuitive physical interpretation, and we put these all on a scale relative to what is possible for a given soil type and climate. To define soils with similar inherent properties and therefore similar soil health potential for the purposes of sampling, we developed an approach to stratifying SSURGO map units based on activity, texture, and drainage classes, which were derived from soil taxonomy, as well as classes of effective precipitation and mean annual temperature derived from PRISM. For more details on this work, please see Vance Almquist's talk here at NCSS. Finally, to estimate soil health potential within soil strata, we look to reference states. For our purposes here, these simply refer to perennial cover types. We expect there to be variation in soil health among reference states due to factors such as the relative abundance of different plant functional types, past and present management conditions, and a residual edaphic variability not explained by this stratification scheme. We are currently implementing the soil health targets concept in Texas and Arkansas in 4 MLRAs that are important for cotton production. Soils within the Gulf Coast Prairie MLRA in Texas formed in clay to loamy, marine, and fluvial sediments, whereas in the Blackland Prairie in Texas, they formed in calcareous mudstone and alluvium, as well as limestone. All the soils in the MLRAs we're working in Arkansas are formed in alluvium of varying particle size. Within our taxonomically based stratification scheme, the soils that are most important to cotton production within the MLRAs in Texas are moderate to high activity, fine texture, moderately well drained. These are all vertisols. And then in some of the coarser parent materials, we have mollisols, which are differentiated from the vertisols here in our scheme, mainly by texture. In Arkansas, we have soils of varying suborders that are low to moderate activity, medium texture, and poorly drained. We have some variation in drainage, so we have another important stratum differentiated just by its drainage status. Within those soil strata that are important for cotton production in the different MLRAs, we sampled a wide range of different reference states. So we considered forests, pecan orchards, rangelands, pastures with introduced grass species, fence rows, hayfields, lawns, roadsides, a wide swath of different perennial vegetation types. Within and across the MLRAs, we also sampled a wide range of cropping systems, varying in terms of crop rotations, tillage practices, and the use of cover crops. Whenever possible, we would sample soils from reference states and cropping systems that were as close by as possible. Pairing is not necessary to implement the soil health targets approach, but it serves as an extra check on our pooling of data within soil strata. Between the paired sampling locations and individual isolated sampling locations, we have at least 40 unique locations within each of the 4 MLRAs. And at each of these locations, we're collecting soil samples for soil organic carbon stocks and texture from 0 to 15 and 15 to 30 centimeters, as well as for soil health indicators for the upper depth interval. We're looking at available water holding capacity using intact cores, slaking index using a smartphone app, 24-hour respiration, and pH, electrical conductivity, and sodium adsorption ratio. Soil sampling and lab analyses for this project are still ongoing, but we can illustrate trends that we would expect to see using data for soybean producing sites sampled as part of the North American project to evaluate soil health measurements. From top to bottom, we have respiration, available water holding capacity on intact cores, and aggregate stability. We see that we have mean soil health indicators for reference states reflecting differences in inherent soil properties and climate. This suggests that we have possible differences in maximum soil functioning from site to site. Another important finding for these soybean producing sites is that we find that under some soil health management systems, some of the functionality of soils may start to approach values observed under reference states even without full perennial cover. We also find that we generally have greater variance in soil health metrics for reference states than we do in row crop systems, suggesting the need to have greater sampling intensity in the reference states to better constrain those estimates. I'd like to conclude with some of the questions that we hope to resolve through this project and subsequent projects. Do our previous findings, for example, of geographic differences in reference state soil health indicators, hold true for cotton producing soils? How broadly should we define soil strata to improve scalability and reduce variance of estimated reference values? How will producers integrate soil health targets into decision-making and development of soil health management systems? Thank you very much for your attention. We would also like to thank the Walmart Foundation for their financial support. And finally, thanks to the landowners and operators who have participated in the study, our team of summer interns, Quanteria Randall, Robert Thomas, and Brent Thomas, and our institutional collaborators throughout Texas and Arkansas. Thank you very much.